One of the long-term goal that Mobilize has established is qualifying an endpoint, registering an endpoint that would be used in an intervention trial to potentially register a new medicine. So who better to tell us about this and kick off the session than someone who has done it already? So Professor Laurent Servais was part of the team that registered the stride velocity 95C for patients with Duchenne's muscular dystrophy, and he's here to tell us about the journey.
Thank you so much, and thank you very much for inviting me. I feel very honored, a little bit stressed, very special. I have a shirt, that's a sign. So my name is Laurent Servais. I'm professor of pediatric neuromuscular disease in Oxford, and that's not where my accent comes from. I'm basically from Belgium. I've been told, okay, just skip your introduction, because this audience doesn't need to be told that the beauty of a digital biomarker is that we allow the simple point tyranny, the simple point, the simple assessment at a certain day in the clinic. With the digital outcome, we can measure patients and get the area under the curve. So I think that you are all convinced of that. You're all convinced of that the digital biomarker is just the sense of history, right? And the regulators have understood that, and FDA has created the Digital Health Center for Excellence, and we see an increasing number of digital outcomes in several trials. These are recent papers. So I understand I am preaching in a choir. But as Michael Moore said, I may be preaching to the choir, but the choir needs a good song. So I'm going to be slightly provocative with you guys. There are plenty of research about digital biomarkers, and this is a recent review of the different digital outcome and wearable device in the field of rare diseases. And you can place virtually your sensors everywhere on the body. But at the end of the day, how many wearable devices and digital outcome are used as a primary outcome or are qualified as a primary outcome? There is a huge attrition rate between our research and between the actual qualification. So I think that, and again, I'm going to be provocative, so please apologize for that. I think that part of the problem comes that we receive as a physician or scientist a beautiful Fitbit or an Apple Watch for Christmas, and we ask the question, what can I do with that? And the day after we go running, and as we run clinical trial, we want to use this new technology in our clinical trials. The short answer is probably nothing, because most of these devices are not fit for purpose. Most of these devices are actually done to help people to exercise, to get motivation, to lose weight, to exercise, but not so much to measure precisely something that can be entered at the end of the day in a database and be supportive of the approbation of a medication. So what we did is, it started from a dream. This is the dialogue between the engineer and the doctor. So the engineer was ready to offer us the moon, and we as doctor, we just wanted an alter of movement. I wanted to be able to identify all the movement of the patients one by one. My patient is moving, I want to know it. And then I want to measure it. I want to quantify every single movement one by one in an uncontrolled environment during 2 years without shift over this time period. And it needs to be a medical device, so we need to have an entire transmission of data security and being compliant with all possible regulation. And then the engineer said, are you sure the moon is not enough? And I think he was right, because we started actually 14 years ago with the first prototype to identify what were the sensors and what we wanted actually to record. Then we made a second prototype that was awful and Bluetooth, and then we made the third prototype that was terrible, and then we made a fourth prototype that was a medical device that was ugly. And then finally, and I'm going to explain you, we gathered the qualification as a secondary endpoint at the European Medical Agency. And then we made something that was a little bit better designed, and then we gathered the qualification as a primary endpoint at the European Medical Agency. So that's how the device looks like in terms of size. And I'm going to present you data that have been acquired with this device. And I need to say I'm not part of this company and I have no financial interest into the device. So that's the size of the device, 25 grams, in comparison with an Apple smartwatch 35, and dimensions that are pretty okay. So the question is, okay, we have our alter of movement, that's fantastic. If I record the patients during 2 weeks, I'll get a mountain of data, right? And the question for me as a physician is, what can I do with this mountain of data? And I can do actually plenty of things, but I need to choose. In terms of symptoms, what is my interest? Is it the power of my patients, the fatigue, the activity, the falls, the ataxia, the abnormal movements? And why? Why do I need to record all of this? Is it to get a signal of action, to be sure at the very early phase that my drug works or not, that I have something objective that helps me to move to a phase 3 trial? Is it in a phase 2 trial to finally demonstrate that my drug works? Or is it to convince the payers or the patients themselves? So a lot of questions, and at the end, the holy grail is a single measure that you can enter into a database and that you fully understand. And for the stride velocity, after several years, for Duchenne, it was the stride velocity 95th centile that we identified. So that's what we can record. Okay, that's a patient's walking. My patient falls. I can measure the strides. I can measure the time that my patient spent raising from floor, and I can reconstruct the walk. So I can identify every single step of my patient. I can measure every single step of my patient. So my patients record during the day, data go in the cloud, and then I've got every single stride. I can measure every single stride. I can have a distribution of the velocity, and I'm going to be interested by the 95th centile. Why the 95th centile? Because I'm a physician, and I know that my patients, the first thing they lose is the top performance. It's not the median that they lose at first, it's the top performance. And that's true for 99% of the diseases. Then you know the problem of actimetry, right? The problem of actimetry has been illustrated in 2 theorems. The first one is when I was in Paris, when the French are on strike, they walk more, right, because of public transportation. And then I moved to Oxford, and when it pours, British people walk less. When it simply rains, they walk more. My 2 theorems. So it means that on a day-to-day basis, you may have a variation, right? But of course, if we average on 3 days, 9 days, you decrease the variability. So actually, the variability of your assessment depends on the time during which you assess and during which you compute. And the beauty in Duchenne is that if you record your patients during 180 hours, which is here, okay, we can see that we have a variability of only 3%. The variability of the ICC that we have today in Duchenne for the stride velocity 95th centile is 0.96, 96, right? So it is it's above whatever we can do in the clinic, which means that in an uncontrolled environment, we can be much more precise than in the clinic because we have a continuous assessment. And then how did we define the stride velocity 95th centile? Not only with the clinical perspective, but also as the most sensitive measure in these patients. And so what we computed was actually the effect size according to the percentile of the stride velocity, and we got the best measure for the 95th. Why not the 99? Because then you consider a certain number of strides that is quite small, and you are much more sensitive to outliers. Then all the questions that we had to answer was, well, does the compliance affect your measure? Short answer is no, compliance does not affect the measure. Do you have controls? And how do you discriminate between controls? Well, that's one of the questions we had. How do you correlate with the gold standards, the North Star, the 6-minute walking test, the 4-step climbing test? But more importantly, what is the added value of your measure? And if you follow a cohort of patients, and your patients are supposed to do worse, are you able to measure it and to measure it faster and more accurately than with another measure? And if your patients are supposed to do better, are you sure that you can actually measure that? And if you measure something, what is clinically relevant? What is the MCID? And this is a very important question, especially for the EMA, much more than for the FDA. But this question of the MCID is a super important question, and that we had really to deal with. Of course, the separation between controls and DMD is important, but then at the end of the day, something that EMA was really, really happy to look at was the sensitivity to change, positive change and negative change. We could demonstrate that if we follow a cohort of patients with the 6-minute walking test, which was the standard, we needed to see a difference and to illustrate a drug, measure drug that stabilized the patients. We need 112 patients during 1 year, which is simply a measure of the effect size. If you want to do it with a stride velocity 90th percentile, you can do it with 14 patients in 6 months, which means that then you demonstrate your added value. And you demonstrate that you can actually do with such measures with 7 times less patients and twice shorter what you do with another gold standard. So that's the long and winding road of the regulatory discussion. We started back 2016, and it took quite a while, because 2016 to eventually get the approval in 2023 as a primary endpoint, we were quite slow. But I think that also the regulators were a little bit discovering the brave new world of digital biomarker and the process of qualifying an outcome measure. And I hope that today we would go slightly faster. So we get the secondary qualification in 2019 and the primary qualification in 2023. And then three things happened. The first one was the this qualification. Then soon after we could publish in Nature Medicine our results. And soon after, this trial from Sarepta, which was highly expected by the field, failed to demonstrate in a gene therapy a difference between the two groups using the North Star, but using this digital biomarker, we could. And that makes a massive difference, right? So I'm Belgian, that's my reference. When something goes well and starts going fast, it's the rocket with which Tintin went on the moon. So now we're working with 30 sponsors in 10 conditions. We have over 2 million of recording, and the conditions we're working on are Duchenne, of course, but also several neuromuscular conditions and non-neuromuscular conditions, including Angelman. And I think that for those of you who don't know what is Angelman, just consider a kid with very severe intellectual disability. How can I assess this kid with my hand and my eyes of physician? If I can just give him a sensor and observe what's happening in the real life, then I have a way to evaluate a kid that I will never be able to do as a physician during a point assessment. So I think that one of the major feed for the digital biomarker is certainly the neurodevelopmental aspect, in which we are very, very bad today as physicians to evaluate our patients. So my take-home message is that now we have made our way in the regulatory landscape, and I hope that all the pitfalls and byways we went through could be useful for others, because this has the potential to reduce the size of the clinical trials and the number of patients and the duration. So how to accelerate the development? With my team, we got 2 umbrella protocols, ActiMocap and ActiLiège. These are 2 different protocols, and we can virtually include in this protocol any patients with any disease. In ActiMocap, what we want to do is to prove that what we measure in a specific disease is exactly what happened in the patients, which means that we measure with a magnetoinertial sensor and with a motion capture to be entirely sure that what we call a stride of 93 centimeters is actually a stride of 93 centimeters. And then, if we are happy, we can move to an uncontrolled environment. Can we really measure in an uncontrolled environment what we do measure in a controlled environment? What is the compliance? What is the reliability? What is the external validation? What is the optimal recording duration for this specific disease? And what is the natural history study? So that's actually what we do. So if I want to share with you the 4 things I've learned over the last 12 years, and this is slightly provocative here, but sensor is key. I think that to understand what you measure and to understand the reliability of your sensor is key. data is key. We need data, and to acquire data is expensive. Having a long-term plan for development of the outcome in a disease is key. What do you target actually? Do you target a qualification with the EMA, with the FDA, both? If you don't care about qualification, do you target to be a very good surrogate biomarker? Do you target to be an outcome for a phase 3 trial? You need to figure out what is your final objective. And finally, the interactions between physicians and engineers is key. And during years, it was sometimes difficult for us to understand each other, with sometimes the engineers speaking a word that we as physicians could not understand, and with our simple words of physician was sometimes difficult to address and to assess. This is a joke, but not so much. And to be honest, this dialogue is sometimes difficult, and to build a long-term relationship between a team of engineers and a team of physicians is key. And I think it's the perfect transition to acknowledge the role of the team of physicians, which is mostly my team in Liège, and the team of engineers from CESNAF. Thank you so much for your attention.